Yes, greetings, Classy. Thank you so much for your time and tuning on to Metric Hub. I'm welcoming you to this one-stop channel, Metric Hub. That is the one channel of your choice. Today we wanna discuss and work on biology. I've seen quite a lot of comments and requests about biology. We wanna look at Botswana General Certificate of Secondary Education Biology Paper 2. So what is happening here is that we have got a paper that we just gonna work together right now. So as you can see, this is a paper 2 biology paper, which is actually set from the Ministry of Education, and it's a national examination. So what is happening here, you should understand again that it's a 2023 paper. So now, without wasting any time, I want us to go straight into the paper. Instructions, you always know that learners, there is always instructions in an examination. So you are not supposed to start writing or start doing anything without answering the question, without reading the instructions. So we have got our content, our questions that are here. The first part is just so contextual, and we are just going to start on question C, which want us to say, to start on question C. Zonation occurs in mangrove ecosystems. We want to look at mangrove ecosystems. So the question is saying, state what is meant by the term zonation. What is meant by the term zonation? Uh, so what we need to understand is that zonation is basically, we can say zonation is basically the horizontal bending of different species up and down the shore caused by exposure, or caused by exposure time, wave action, and the salinity of changes. So what you need to understand is that there is such an experience, which we are saying horizontal bending of different species up and down, the bending of species up and down, and we are saying again that up and down the shore, which is actually caused by exposure time and also wave action and salinity changes. We are looking at an ecosystem here. So now other terminology or other definitions might want to define it as we're gonna also put it in other ways or in other definitions, as that is actually the distinct distribution of different mangrove ecosystems. Uh, Let me just put it in writing now. We are saying it is basically, we can also say that it is distinct distribution, distinct distribution, distinct distribution of different mangrove ecosystems, different mangrove ecosystem, uh, distribution of different mangrove ecosystems, or we can say species, the species that are in the mangrove ecosystems or communities, or a community, which we say that or communities, or communities which actually along a gradient of environmental factors, along a gradient, along a gradient of environmental factors, environmental environmental factors. So these factors, we can just name them. We can say environmental factors such as salinity, which we are saying mangrove species have varying tolerance to salt concentrations, influencing their distribution. So in terms of mangrove species, their tolerance to salt concentrations, it is actually varies, which actually affect their distribution. We can also talk about waterlogging, which also some also species are adapted to prolonged flooding, while others prefer better drained areas. So I'm saying the varying factors or environmental factors that we are looking at. We are looking at salinity, we are looking at waterlogging, we are looking at soil type, which also specific soil, we need to look at specific soil types such as muddy or sand substrates at the same time as tides. So we are looking at these as factors that actually affect the communities and distribution of mangrove species along a gradient of environmental factors. So I'm saying example, e.g., the environmental factors that we are talking about. We are saying soil type. We can say salinity. We can say salinity. We can also say otherwise. We can also look at waterlogging, which I'm saying all these factors, ne, they are so varied, which actually affect the distribution of what of mangrove species. Each of them, the species are tolerant to different conditions. So these conditions affect their distribution. So distinct distribution of different mangrove species or ecosystems or communities along a gradient of environmental factors such as soil type, salinity, waterlogging, and tides. So identify the zone in which plant A is found in this ecosystem. So what happens is that we're saying the plant A where it is found, identify the zone in which plant A is found in this ecosystem. Uh, it is actually found in the splash zone. Plant A, it actually have some type of a plant which can actually be found on a splash zone. It can be found on the splash zone or upper littoral. You can say splash zone or it can also be called upper littoral. Upper littoral can say it is also called upper littoral. Uh, and also we are saying only weighted by the spray or spray of only salt and tolerant lichens survive this one. So we are saying the environment is actually salty, and then it is actually surviving. It can only survive lichens that can survive there. So in other words, what you need to understand is that it's a splash zone upper little, which we are saying again that only weighted by spray to say that there is no much water, only weighted by spray, only weighted by spray. So in other words, we are saying so only salt, only salt. So now what we are saying is that that's what is happening there. So now state one natural and one man-made threat to ecosystem named above. The man-made and natural threats. In terms of natural, we are looking at factors that affect uh, a seaport size ecosystem. So now what we are saying is that it is affected by what? It is affected by factors such as one, in terms of the natural, we can talk about storm waves. Natural, we can talk about storm waves. Storm waves can be natural. Storm waves, which we are saying that it actually rips, which has got an effect on what on rip. We can say that it rips off it rips ripping organisms off, which can actually rip organisms off, or it can rip organisms off. We can also talk about sand burial, storm waves. or sand barrier, sand barrier, smoothing, sand barrier, which we are saying smoothing, what rock pools, smoothing rock pools. So now, what we need to understand is that these are natural factors or threats that can affect our ecosystem that we are referring to in terms of many made threats to the ecosystem. We can look at we can talk about tourist trampling, the walking of tourists. When tourists moves in there, they actually trembling, and also when they are collecting shells, trembling and collecting trembling or collecting shells. We can say also that one. We can also have another man-made as what as oil spills. We can talk about oil spills, oil spills from passing ships, spills from passing oil spills from passing ship. We can say the ships also, as they pass, they can also spill their oils, which can also affect our our seaside ecosystems. So tourists walking and also collecting shells, they affect the ecosystem in there. And then oil spills also, when ships are passing, they also have an effect, which we are saying that we can also identify them as what as many-made and natural many-made and natural what natural threats to the ecosystem. And then state two reasons why this habitat should be conserved. The importance of conversations. What are the importance there? One, we can talk about the the reasons for conversation. We are saying one, the seashore must be conserved because it is a necessary area for small fish. One, it provides necessary area. It provides necessary area for juvenile fish. We are saying juvenile. We are saying juvenile is just say small for juvenile fish. The small fish can actually find territory or habitat there, and also we are also supposed to understand that it actually which actually supports fisheries. In other words, we are saying supporting fisheries, and then we can also talk about another point which has to do with natural coastal armor. It also provides it is a natural coastal natural coastal armor, which we are saying again that it reduces erosion and storm damage. Therefore reduces erosion. It reduces erosion and storm damage. It reduces erosion and storm damage. So these are the reasons for the habitat conservation. And then state one method that can be used to conserve this ecosystem. How do we conserve it? Conservation method. We are saying what can be done. This can be done. One, we are saying create marine protected areas. Create marine protected areas. creation creation of marine creation of marine protected areas. Marine protected marine protected areas. Marine protected areas can actually help there. And then we can talk about rope off sections. You can actually rope off or barricade rope off rope off we can talk rope off sections where certain areas must not be entered, and no harvesting, uh, avoiding no harvesting, no harvesting. And therefore, we are saying if there is no harvesting, at the same time, boardwalks to keep feet off fragile, no harvesting or a boardwalk. So you see what we are saying is that creation of marine protected areas and rope-off sections, that there are some areas which cannot be, which people cannot actually get in. So in that regard, we are saying it actually helps to conserve the ecosystem. We have got this. We have got the next question, which actually talks about the blood. Question below is about blood, and then complete the table using a tick. We are supposed to complete the table using a tick, and then what is happening is that we need to mark true or cross as false to show if the statement applies to red blood cells or white blood cells. So we have got an example that has been done. So we have got statements on the first column, red blood cells and white blood cells. So now what happens is that the numbers may increase following infections. The numbers may increase following infections. Red blood cells, their numbers does not increase following infections, because by nature we are saying what is the function of red blood cells. What increases is what is white blood cells, because they are there to fight against illnesses. They are there to do to provide an armor to the body. So what we are saying is the white blood cells are the ones that increases, because they are there to provide protection to the body. And then transport oxygen. Red blood cells are there to transport oxygen. They basically transport oxygen. So what we are saying is red blood cells, they do transport oxygen, and white blood cells, they don't, white blood cells, they are not there, they don't transport oxygen. We are saying red blood cells are the ones that transport oxygen. Contains the nucleus. What happens is the red blood cells, they don't contain a nucleus. Red blood cells do not contain a nucleus. We find a nucleus on what? On white blood cells. White blood cells are the ones that does have a nucleus, and then he has a biconcave shape or a disc shape. The disc shape, we find it also on what? We find it on red blood cells. Red blood cells, they are disc shaped. They have got a biconcave shape, which actually helps them to perform their function, and then the white blood cells are not. And then the next question, engulf bacteria. Engulfing of bacteria is just to say that the we find it happening in the white blood cells as they destroy the disease-causing organisms. They engulf. They actually gather around. So red blood cells do not engulf bacteria. So bacteria is destroyed by what? By the white blood cells. That is what is very important. That is what you need to understand with regards to the functioning, the characteristics of blood cells. We have got people who are asked to donate blood during blood derives People who are often asked to donate blood during blood drives in the community. We understand that the blood collection group. So what you need to understand is that blood donation is very important. Blood donation is quite important, and we need to know that it is very important that people also give blood in order to save lives. I'm sure we see them in schools, we see them in public places. So now, what we need to understand is that blood donation is quite important. And then, where is the blood stored? Where is the blood storage? Where is the blood stored? Blood, when it is donated, class, it is there is what is called blood bank refrigerators. It is stored in blood bank. It is stored in blood bank refrigerators. These blood bank refrigerators, what happens is normally they are kept at 4 degrees Celsius, which are at 4 degrees Celsius, which actually helps to keep cells alive for something like 30 to 30, 35 to 42 days. So what happens is that blood bank, I mean, when blood is donated, it is classified and, and kept within the blood bank where it is going to be retrieved whenever it is needed. So it is actually stored at within a blood bank for it to remain usable and useful to people by being within a refrigerator which is normally around 4 degrees Celsius, which can help to keep the cells alive for something like 35 to 40 42 days. And then why is it important? Why is it important to have blood, to have stored blood? Blood storage is very important in order to save lives. So we are talking about saving. It's actually important in order to save trauma patients or to save trauma victims that are suffering trauma. They need blood addition. They need to be transfused with blood. We talk about anemia patients. We talk about anemia patients also. Anemia anemia patients also, they need blood. You can talk about surgery. It can also be used in surgery. Surgery as well. So now, uh, so blood is basically a life saving. It is actually, in other words, we are talking about saving lives. Uh, saving saving lives. Lives must be saved. It's a life-saving fluid. Blood for it is kept in order to save lives. Saving lives is basically the main reason why we have to have blood stored. So we need to understand that when every time blood is needed is readily available, and then state which group is known as a universal blood donor and give a reason for your choice. Universal blood donor, in terms of blood grouping class, it is very important that we understand that blood is classified with, we have got different types of blood grouping. We have got ABO blood grouping and also rhesus factor blood grouping. So now, the universal blood donor, we look at a blood that when it is given to any person, whether blood A, blood AB, blood O, blood B, that person can be able to survive. When a patient is given a blood type that is different from his or hers, what happens is we have got blood clumping, which actually causes the blood to clot, which would result in what? In blood clotting, which will actually cause arteries and capillaries or blood vessels clotting and blockages, which at the end of the day might result in what? In problems such as we can talk about paralysis and any other serious conditions which would eventually might cause death. So universal blood group is blood group O. It's actually universal blood group is actually blood group what? Blood group O. We are saying blood group O, blood group O. It is universal. The reason is that is actually blood group O is actually universal. What happens is because it can mix. Reason is because it can easily mix. It can easily mix with other various blood types, with other blood types without causing problems, without causing blood clotting, without causing blood clotting. So now, blood group O is actually universal. We say that they are other blood types, they've got antigens on their surfaces. In other words, we say that blood O does not produce, it does not produce any antigen, does not produce any antigen. Antigen is a substance which, whenever it is introduced in the body, it results in the production of antibodies. So what we are saying is that blood group AO does not produce antigens. It has no antigens that it produces. So being like that, it produces no antigens, class. It has no antigens that it produces. We are saying it does not produce any. It does not produce any antigen, so cannot cause any problems. Cannot cause any problems in the patient's body or to the receiver, to the blood recipient. Cannot cause any problems to the patient. You understand? So therefore, blood O has no antigens. It therefore does not have is not recognized whenever people receive blood that is opposite from theirs. They are blood type A and blood type B. Blood type A and B, they produce their own type of antigens, where A produce antigen A, B produce antigen B. So when it is also A, B, it will produce antigen A and B. So in other words, we are saying when it is identified as a foreign substance in the body will start to produce those antigens, which will cause problems, destroying of those blood cells, and then result in what? In clotting. So in other words, what you need to understand is that blood type O does not produce any antigen, hence cannot cause problems to patients when it is given to them. And then we have got the next question, which has to do with Julie need blood transfusion. Julie is in need of blood is in need of a blood transfusion. She has blood group B. Describe the possible effects of giving Julie blood transfusion with the blood group A. Uh, the effects blood group A will be identified will be identified or recognized will be identified by Julie's body Julie's body as a foreign substance which can which can be destroyed, which can be destroyed by antigens from from her body, which can be destroyed by antigens produced by a blood type blood type B. So this is what happens is that blood type B, I said that you guys, what you need to understand is that blood type A, it produces antigen, antigen A. Blood type B, it produces antigen, antigen B. So each blood group produces the antigen that is exactly of its own. So if a person is given an opposite blood type, her body or his body will produce or will start to fight and destroy the blood type that is opposite the patient has been given, which results in conditions that we are saying that the blood capillaries or the blood veins will clot because that blood that is given, which is opposite, will be destroyed. So we have got this question here, which we are given that the graph shows the mass of 3 different air pollutants released into the atmosphere each year from 1990 to 2010. So we are given that question there. So now what we need to understand is that on vertical axis we are given mass released in each year in tons. We are talking about what? About pollutants that are produced. So now we want to see these questions and then answer them. We want to see what is happening here. So we are saying we are given the key here that the first one is what is carbon monoxide, the second one is sulfur dioxide, the last one is what we are saying 1, 2, and then 3 is the nitrogen oxide. So these are the different gases that are the different pollutants that are given or which has been produced from 1990 to 2000 to 2010. They are the different pollutants that are produced there. So now the question is saying define the term pollutant. Define the term pollutant. This is a substance, or we are saying it's a substance or agent. It's a substance or substance or agent which we are saying that is released into the environment, that is released. What happens is that it is released into the environment. It's released into the environment and can cause harm to living organisms. Causing harm to living organisms when it is released into the environment, it causes harm to living organisms, ecosystems, or human health. Organisms ecosystems or human health. So now what we need to understand is that the pollutants basically most often, or we can say they can be chemical, they can be physical, they can be biological in nature. So what happens is that these substances, when they enter into the environment, they cause harm to living organisms. They can also destroy ecosystems. We can also talk about them also affecting human health. We have got respiratory diseases that people can suffer. We can talk about substances which, when they get into the environment, ecosystem are destroyed, habitats, living and animals, plants and animals are destroyed. We talk about these things quite a lot. Even the formation of acid rain that destroys our ecosystems is a typical example of pollution, which is because of the greenhouse gases that are emitted into the atmosphere, resulting in acid rain which at the end of the day can affect our what? Can affect our plants and animals. Identify the air pollutant that showed the smallest change in the mass released into the atmosphere from 1990 to 2010. So this, we are just looking at the air pollutants that are given in question, and then we need to see which one shows the smallest change in mass released into the atmosphere from 1990 to 2010. So what we are looking at is that 1990 carbon monoxide in 1990, we are saying it was something like uh, five this is we said what, 500 tons, which can be, we said it's 500 and 550, 60, 70. So this can be 500. So each line represents it represents two. So we are saying it is 500, uh, 520, 540, let's say 500, we can say 500, 545. And then that was carbon monoxide. I mean, carbon monoxide 20, we are saying in 20, in 20, 1990, 1990 carbon monoxide, we have got 1990, we have got 540. We can just say 540. In 2010, we have got something like it is something like 200 and 200, 220, 240. It can be 240. And then we look at sulfur dioxide. Sulfur dioxide, it was 280, 1990, 280. And then in 2010, it is when it went to 160, 160. And then we look at nitrogen oxide, it was 90, 93, 92. And then it went in 2010, it went to uh 20, 40, 60. It went to 60. So now what we are saying is these are the changes class. We are saying 1990 and then 2010. So you are looking at these changes. You are looking at these changes, then you can be able to calculate the differences. So the one that shows a smallest change, the one that showed the smallest change in mass release in the atmosphere, we can see the variation. Here it is almost half. It is almost half here, the change. And also 180, 280 to 160 is quite big. 90 to 60 is something like 30 something. So what we are saying is nitrogen oxide is the one that showed nitrogen nitrogen oxide showed a what? Nitrogen oxide showed a small change. We are saying we are looking at the figures. We are calculating the variation of the figures. from 1990 to what, to 2010. So identify the air pollutant from which 360 tons was released. We are saying 360 tons was released into the atmosphere in 2000. So you are just going to the year 2000 and then you look at the air pollutant that is got 360 tons. 2000, 2000 here in the year 2000, our year 2000, we are saying where we have got 360 in 2000. This one, it is 90, it is 91. It can be this one, sulfur dioxide. Sulfur dioxide is 240, and then we can go up. The next one is carbon dioxide, carbon monoxide, which is carbon monoxide. It gives us what? 360. 360. So the 360, we are getting it on what? On carbon monoxide. Carbon monoxide, it has got is carbon monoxide. Carbon monoxide, like I'm saying, it is actually given there. It is actually given. We can see it. It is actually given that carbon monoxide, it has what? It has 360 tons in 2000. So it is just given on the graph. We get it from the graph. Calculate the change in the mass of sulfur dioxide released into the atmosphere. Change in the amount of sulfur dioxide released in the atmosphere 1990 to 2010. So we just need to say they calculate the change in mass. So the mass is given from there. We are saying sulfur, we calculate the change in mass in sulfur dioxide 1990 to 2010. Sulfur dioxide 1990, sulfur dioxide 1990. We are saying 1990, it's what is 280. 1990, we have 280. And then 2010, sulfur dioxide 2010, we have got 100, 120, 140, 141, 140, 141, 141. So now we are saying 280, 1990, we are saying 1990, we have got 280. And then 2010, we have got what? 2010, we said we have got 141. 2010, we have got 141. So calculate the difference. Difference is equals to less. What are we lessing? 280 minus 141. And then we get our answer is there. 80 is there. 8 minus 5, 3. So we are saying our answer is what is 38. What is the question saying? The change, calculate the change in mass of sulfur dioxide released in atmosphere 299. So we are saying the change is equals to 38. We are saying is 38. What is they are given in what the units? 38 tons. The change is 38 tons. 38 tons is what is the change from 1990 to 2010. Sulfur dioxide decreased by 38 tons. It decreased by 38 tons. That is what is given in this case. It decreased by 38 tons. So that is what you need to understand. So now let's check the next question. What is the next question saying? List two. The next question is saying list 2 reasons for the change in the mass of sulfur dioxide released into the atmosphere in that year. List 2 reasons why the change was that. We are saying what can be the reasons for the change. Reasons for the change can be because people switched from coal to solar and natural gas. change in the form of change, the changing of the source of fuel. We are saying sulfur dioxide dropped. Reasons for the drop of sulfur dioxide. So list the reasons for the change. So we are saying the change is what? The drop or decrease in sulfur. We are saying the change or decrease in what? In sulfur dioxide. The change or decrease in sulfur dioxide. So now, what is the reasons? We are saying as people might have switching. We are saying switching from coal to solar, switching from coal to solar. People switch from coal to solar and natural gas. So the form of the source of fuel changed, or people changed the type of fuel. They have to use some natural, environmentally friendly sources of energy. The sources of energy changed from what? From coal to what? To solar and natural gas. Therefore, the decrease might have been there. And then we talk about also other reasons, such as uh, we have got other reasons that might also be there, which can be because of what can also because it can also because of gas desulfurization scrubbers in factory chimneys, uh, gas gas desulfurization, gas desulfurization scrubbers, the scrubbers which can also maybe in factory chimneys, which might have been put scrubbers in factory chimney also might have caused a decrease in the amount of sulfur dioxide that has been produced. So what happens is that this all have been to do, they have everything to do with the decrease in what? In amount of sulfur that has been produced. So the desulfurization scrubbers are there to reduce the amount of sulfur that is produced from factories and industries, which we are saying that it can also reduce the amount, or it can cause the decrease in the amount of what? Of sulfur dioxide. Another common air pollutant produced from man's activities is carbon dioxide, which is right. And then name the key environmental problem caused by this pollutant. Uh, the sulfur dioxide problem that it can cause, we can talk about global warming, global warming. We are talking about the global increase in temperatures as a result of temperature. I mean, as a result of greenhouse gases, which actually could also result in what? Which can also result in climate change. Uh, which can also result in what? In climate change. Climate change might be experienced as a result of global warming. We have got climate changing. Instead of being our summer times being from this month, being for 5 or 6 months, it's now stretching more than usual. So what happens is that global warming becomes a challenge, which actually affects our ecosystems. And then state two adverse effects this problem has on the abiotic environment. Don't forget we are talking about on non-living. We are saying factors that it has on non-living organisms, or non-living elements of the ecosystem or of the environment. So we are saying abiotic. We are not saying, we are saying non-living elements in the environment. So what we need to look at is that what are the effects of this problem? We talk about the ocean acidification. One, we have got ocean acidification. the ocean becomes acidification, which we can talk about, which can also result in coral skeleton uh, dissolving, dissolving coral skeletons. And then we can also talk about melting permafrost, the melting of permafrost, melting permafrost, melting permafrost, melting permafrost. It actually releases. We are saying release more methane, accelerating the problems to do with what with coastal flooding. So releases more methane, and more methane gas can be released. More methane can uh, can be released, which we can also give as also problems that can also arise there. There's also problems that might also arise in this case. So we are saying melting permafrost, these are like we are talking about zones that are temporarily frozen. So now what happens is that when the temperatures increases, they can also result in earth movements that are various that we can talk about. And also what happens is that these permafrost and also we are saying the problems can also result in coastal flooding, where areas close to the sea can also suffer from floods as a result of the melting of what of frozen layers of what of the glaciers and so forth. We have got this diagram there that represents the human digestive system and associated organs, the human digestive system and associated organs. So we are given here that this is it. We need to identify the different organs that are named here. It is the digestive. We want to look at digestive structures. So the question is saying name the structures labeled PQR. Name the PQP. What is P? P, we can see that P is the liver. P is the liver. Q, we can talk about Q as what? As the gallbladder. Q can be the gallbladder. And then we can talk about R. R can actually represent uh, R can actually represent the small intestines. R can represent the small intestines. This can be small intestines. And then identify complete the table by writing the letter next to the structure where function occurs. We are saying absorption of nutrients into the bloodstream. We have got absorption of nutrients into the bloodstream. It also takes place in the what? In a letter which is R, and then production of bile. We're saying bile is produced from the bile is produced from from the pancreas, which is associated with the liver. Can say, let me just say Q, and then digestion of proteins. Where do we have digestion of proteins? Remember to say, class, that we need to be able to understand this. What happens is the digestion of proteins in the human digestive system, digestion of proteins in human body, digestion of proteins in human body. What happens is that sometimes you find out that this diagram is sometimes you might miss it because you are not able to see it properly. You should be able to you need to be able to see it. You need to be able to see it properly so that you can be able to get it right. So when we look at this, we are saying it actually, the protein digestion, it's a process that is very important also in this, it can actually be digestion. It is actually done by enzymes, which can actually take place in the stomach because of the gastric juices, which actually contain the pepsin enzymes and so forth. We can also talk about in the small intestines, which secrete also some trypsin and also the chymotrypsin carbohydrates, which are further broken down into amino acids. We might also talk about the pancreas, which also produces the digestive enzymes, including those that break down also the proteins. So we are saying the pancreas, the part that is responsible for where digestion of proteins can occur. We can say small intestines, which can also be, it can also be R. We can say R, that is where proteins can be digested in the small intestines. And we can also say it can also take place, it can also take place in the what? We are saying Q can also be the stomach. This can also be the stomach here. We say this can be the liver. This can be large intestines. This can be large intestines, can be small intestines. So what we are saying is Q, we can give it to say that Q can be the stomach. You can say it can be the stomach, liver, and so forth. So now that's how we can define them. Let's just check the next question. It's saying the pancreas is part of the digestive system, and then name an enzyme produced by the pancreas, the food mutant it acts on, and part of this action it acts on, and the product of this action. Name of enzyme that is produced from the pancreas. We are saying that it actually name of the enzymes. We say the pancreas, it produces the pancreatic juice. It produces the pancreatic juice, food nutrient that it also digests. It also digests proteins. Proteins are digested from the pancreas. And then what we are saying is that what happens is that when proteins are broken down into smaller peptides in the stomach, So now we are saying the products, the food nutrient that it acts upon on, it acts on, and the product of this action. We can talk about what the products. We can talk about peptides. Peptides are produced. State state the role of the pancreas and liver in reducing blood sugar levels. The role of the role of the pancreas and the liver in reducing blood sugar levels. So now, what we need to look at here is that we have got, in other ways, even if we can look at these various, uh, these various enzymes, we have got also even amylase, which also is responsible for breaking down starch into maltose. So I was saying there is also, we can also give an example, and I mean another type is what is amylase. What does it produce? What does it digest? It digests what? It the amylase is it digests starch, and then the starch is broken down into what? Into maltose. The starch is broken down into what? Into maltose. That's another example. So we are saying pancreas. How does the pancreas and the liver reduce blood sugar levels? In terms of the blood sugar levels, class, what you need to know is that the pancreas, it has got specialized beta cells. So the beta cells, they releases insulin. Uh, it releases actual insulin. It releases insulin. The pancreas releases insulin from beta cell, release insulin from beta cells, from the beta. So now the insulin converts, the insulin actually, the insulin which actually converts glucose into glycogen. It releases insulin, and then uh, it actually releases insulin, and then the liver also converts excess glucose. The liver, it actually converts, it converts excess glucose. Converts excess glucose into glycogen. I'm sure I remember that this actually has got a lowering effect on the glucose levels. Glycogen lowering the glucose levels. A process which I remember you should understand the glycogenesis. Glycogenesis, that process which we are saying, it is also responsible for what? For lowering of what? Of glucose in the body. We have got microbes are used in the production of many different food items. Microbes are used in the production of many different food items. Microbes are used in production of many different food items. So now the question is, the table shows information on production of two food items. It comes with the production of two food items. So now what we need to look at is, we want to look at what is microbes. First and foremost, I want you to understand what are microbes. That is the first thing that you need to understand. So therefore you can be able to understand. So microbes, when we look at them, we are looking at, or what is microbes? is actually, we are looking at microbes as what? As microorganisms. These are microorganisms, microbes. We are looking at microorganisms, microorganisms. We are looking at microorganisms, and then we are looking at a tiny living thing that can be seen by a naked eye. Tiny living thing that cannot be seen, that cannot be seen by naked eye. We call that a microbe. So now what happens is that they are quite diverse of them and can be found almost everywhere, everywhere on earth. We can talk about them from human gut to even deep sea vents. We can find microbes there. So types of microbes, we have got bacteria, we have got viruses, we have got fungi, we have got protozoa, and then we have got acacia. All these are different types of microbes. So now what we need to look at is that we need to look at this and be able to understand to say, uh, what are these different microbes that we are looking at? What are these microbes, the different types of microbes that are there? So we are saying they are used in the production of different food items, microbes. I want you to understand that microbes, they are actually used in different, they are very useful in various ways. We say that it can be important in human health, which can actually, in terms, they can play a role in digestion, immune function, and overall health. They can also be important in ecosystems, which has to do with the decomposition and also the recycling of nutrients, food production in terms of fermentation, such as bread and so forth. Biotechnology, I'm sure that we have seen that one in terms of genetic engineering and biotechnological applications that we might look at. So now, these are microbes that we are talking about. So the question is saying, identify the food items. We are looking at the food items. We are looking at the food items that are there, and then name of the microbe used in the production, name of the microbe that is used in the production. So now let's quickly just answer that one. So we have got the food item, bread, bread, and then name of the microbe used in the production of this one. The bread, what microbe is used in the production of bread. So like I said, they are quite uses, there are quite a lot of uses of them. So bread, we have got it's called Saccharomyces cerevisiae is the one that is used in what? In bread production. It is actually used in bread production. And then we are saying the group where it belongs, it's a fungi. And then chemical products of microbes used in the production of what? Chemical products that are there. We are saying with this, what happens is that in terms of the bread, what is what is produced? We talk about chemical products of the microbes used in this one. We talk about carbon dioxide. Carbon dioxide. Carbon dioxide is produced. Carbon dioxide. We talk about carbon dioxide as also the one that is produced there, which normally we look at it and then it can cause the dough to rise and become aerated when we are looking in bread production. And then name number second, it's saying it wants us to look at this. What is this one which is used by lactobacillus? So it is also, it is yogurt. We can talk about yogurt. Can talk about yogurt, and then it is actually, we are saying it is lactobacillus. So now, what is happening there is that it is a bacteria. It is a bacteria, and then the chemical product is lactic acid. The bacteria, and then it produces what? Lactic acid. So now, during production of both food items, the microbes carry the process of anaerobic respiration. The microbes carry the process of anaerobic respiration. So now we are saying that there is a process of anaerobic respiration that takes place there. So what we asked here now is that we want to look at this and then define anaerobic respiration. I'm sure you remember when we look at types of respiration, we've got anaerobic and aerobic respiration. I'm sure that we have looked at this. So it is a type of cellular respiration uh, that occurs in absence of oxygen. Type of cellular respiration that takes place in the absence of oxygen. We call that anaerobic respiration. Oxygen is not available. There is no oxygen. So in other words, what happens is that is actually cells generate energy from glucose and other organic molecules without using oxygen. There is no oxygen in this case. There is no oxygen, and then energy, we are saying, is actually generated without using oxygen. So write down the word equation for aerobic respiration. What happens is that the word equation that occurs there is that it's glucose. Glucose is broken down. We're saying glucose is broken down in the absence of oxygen, and then we have got ethanol. It produces ethanol, and then plus what? Plus carbon dioxide. Ethanol plus carbon, ethanol plus carbon dioxide. Glucose is equals to ethanol plus carbon dioxide. So in other words, what you need to understand is that there is no what? There is no oxygen involved. There is no oxygen involved in this case. There is no oxygen involved. So we need to understand that there is no oxygen involved. There is no oxygen involved in this case class. So I'm just giving you the chemical formulas for this. That is what we are referring to. So it is actually a process class that is important to allow cells to generate energy in environmental conditions, in environments where oxygen is not available or oxygen is limited. When oxygen is limited or oxygen is unavailable, we are saying anaerobic respiration is actually allowing cells to survive, to have energy in that kind of an air oxygen limited or oxygen suffocated environment. We have got the information from a nutritional label found one of the food items in Part A, and then nutrition facts serving one container has got 150 grams. In terms of the kilojoules, it has got 585 kilojoules in the amount of servings, and also whatever is given. So the question is saying state the energy content, state the energy content, state the energy content, which is uh, state in kilojoules of 2.5 containers of this food item. So what is happening is that we are given the energy. We are given the energy. In this case, our energy is given to say that we are given that it is 585 kilojoules. So in of two, we want of 2.5 containers. So this is one container. So 2.5, we are going to say that 2.5 multiplied by 2.5 multiplied by 585. 585, we say 2.5 multiplied by 585. That's what we need to get. That's what we are saying. You put it through the calculator, you would find out that this will give you, this is 585 kilojoules. Don't forget anyway. It will give you 1462, 1462, 1462.5 kilojoules. That's the answer. So we are just getting what we are given, and then we multiply it by 2.5 to get the what? To get the kilojoules that are needed there. And then we are given again which food nutrient supplies the most energy. Which food nutrient supplies most energy? We are supposed to look at it to say that we can look at the food nutrients that are given there. Food nutrients that supplies the most energy. We are given the food energies there, the amount of energy which are there. So we can see that 23, which is coming from carbohydrates. We have got 23 coming from carbohydrates, which is the highest, considering every other nutrients that are there. So it is what? It is carbohydrates. It is carbohydrates. Why is this food not? Why may this food not be a health choice for a person with heart disease? Why may it be a problem with people, to people with heart disease? We are saying actually it is actually the problem is because uh, a person with heart disease, we have got it contains high sugar, which can cause obesity. It contains more sugar. Yes, more sugar, which may cause obesity, which may cause obesity. So therefore, we are talking about problems that might arise because of obesity there. So that is why we cannot have to give people with heart diseases, because when they are obese, they are also going to be overweight, which results in conditions such as heart failure, which can result in that condition. Diagram represents the menstrual cycle of a woman, which is given there. We have got the menstrual cycle, which is given there, and then what we need to answer is that this is it, a human menstrual cycle class. This is in this case, we are talking about, it is the endometrium lining. This is the endometrium lining. In this case, we are seeing the developing follicles. which are in, I'm saying the developing follicles. We can see that these follicles class, let's understand that the developing follicles, we must put it from this side, developing follicles. So now, what the question wants us to understand is, uh, it wants us to say, name the structures labeled RST, RST, name the processes that are labeled RST. So in this case, class, what you need to understand is that when you are given this, we can say secondary oxide, and then we can also have at this stage, as you can see, we have got ovulation that is occurred here. Ovulation is occurred there. So we have got the we have got the corpus luteum. So now that is it. So S is the secondary oxide. R we can talk about is the blood vessel. And then we are saying when ovulation occurs, we have got the cell that remains, which was enclosing the egg. So now what happens is that ovulation, after it occurs, we remain with the corpus luteum, which is actually there to produce progesterone, which can also identify that it continues to support the thickening of the endometrium. So now what we need to understand is that we're saying R is a blood vessel. S is a secondary oxide. And then T, we said is corpus luteum. Is corpus. And then in which label structure does implantation occur? Where do we see implantation occurring? Implantation, it occurs on the what? On the endometrium. It occurs, we can say R, which is the blood vessel. We can say it is the endometrium, which we are saying that it occurs on R. Implantation, the attachment of what? The attachment of a fertilized egg is what we call implantation. It occurs at R. So we are given the calendar to answer the following questions. We are given the calendar to mark, to answer some questions here. And then let's see the questions that are coming there. Women's menstrual cycle begins on Tuesday, 1st November. It begins on Tuesday, the 1st of November. Place the X, the letter X on the calendar on each of the 5 days that menstruation is typically expected to occur. Menstruation, we are saying it is typically, we are saying it actually has to occur on days 1 to 5 on days 1 to 5, that is where we can have menstruation occurring. Days 1 to 5, days 1 to 5. So we can say it can be menstruation can occur from days 1, 2, 3, 4, 5. That's where we can have, we are saying time, we are saying menstruation can occur at this time. That's where we can see menstruation occurring. And then the question is saying, place Y, place the letter Y on the calendar on the day that ovulation is most likely to occur. Ovulation, it occurs also on day 14. So ovulation, it occurs on day 14. I'm sure that we understand this ovulation occurs on day 14. So we are saying Y is when we have got what? This represents ovulation date. The ovulation date can be day 14. And then let's see the next question. How many days on average does the menstrual cycle last? How many days on average does the menstrual cycle last? It's the cycle last like averagely the cycle is 28 days on average. And then state a number of hormones. A number of hormones are involved in the menstrual cycle. The concentration of one of the hormones in the blood drops dramatically several days after ovulation if fertilization has not occurred. Yes, the it drops dramatically if after ovulation if fertilization does not or has not occurred. If it has not occurred, state the name of this hormone. The hormone that actually drops is actually progesterone. Progesterone, progesterone hormone. And then explain how knowledge of changes during the menstrual cycle can be used to prevent pregnancy. Uh, how how knowledge of menstrual cycle can be used to prevent pregnancy. We've got people can talk about natural contraception, avoidance of copulation, days 10 to 17. We are seeing the fertile window period uh, can be used as a contraception method uh, to avoid pregnancy. To avoid pregnancy, which we are saying to avoid pregnancy. We are saying natural contraception. This is, we are talking about natural contraception, guys, here. Natural contraception method to avoid pregnancy. And then this actually, we are saying, which is days 10 to 17. Days 10 to 17, which are known as fertile window period, which is fertile window period, which is fertile window period. And then it is also named as what? The rhythm method. The rhythm method. So the method, the knowledge of menstrual cycle, it can actually be used as a natural contraception method in avoidance of pregnancy on days 10 to 17, which are called infertile days. And then people can avoid pregnancy in those days. We can also talk about that to say that people can also, uh, even otherwise, we are saying they can actually avoid, avoid sex. We are saying it can actually, we are saying avoid sex, not pregnancy. Avoid sex, you can say avoid copulation, days 10 to 17, which is fertile window period. And then in these days, that is when a person is highly fertile, and then there are chances of pregnancy. So as you know that ovulation occurs on day 14. So a person can avoid to have uh, uh, copulation, to avoid lovemaking on those days. So what we are saying is that pregnancy can be avoided using the knowledge of contraception. I mean, using the knowledge of menstrual cycle. Yes. So we have got the diagram that shows section of a human skin. It shows the section of the human skin. So we are given the section of the human skin. So name the structures labeled P and Q. P, P. We are saying name the P and what and Q. So this P can be it can be an erector muscle. P is an erector muscle, which is actually, we are talking about thermoregulation in this case class. Thermoregulation, the control or the balancing of body temperature by the skin, some physiological changes that takes place. So now is the erector muscle, and then R is the what? Is the hair? Is that R is just the what? R is the hair. R is the hair, which you know very well that the skin here, it has got the condition of it has got the condition of it has got the condition of stenting or of flattening in order to control the temperature, in order to control the body temperature. So now uh, uh, there we can talk about blood vessel. We can talk about this as a what? Sweat gland, sweat pore. It can be seen as the sweat pore. And then we are saying all these are there to control and regulate. This is the skin layer. Is the skin layer. So what we are saying is that we need to understand this. So now let's quickly look at this and see. State the effect of cold temperatures on P and Q. On P, the 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 effects of this on P. P actually it contracts and has turned on a cold on a cold day. P it actually it is there to it contracts, causing the hair to stand, and then to so that it can trap what? So then it can to trap to trap insulating to trap insulating air layer. I'm sure you have seen that on a cold day, people they develop what are called the goosebumps. The goose the goosebumps. So what happens is on a cold day, hair has to hair it stands up because of the what of the erectile muscle or erector muscle, which actually contracts in order to put the hair to stand upright. So therefore, it can be able to trap the air in there. So R actually we are saying R, which is uh R. What it does is that it actually it contracts, and then what happens R is just it stand upright to trap the air in order to insulate the skin. You understand? And then which term is used to describe what happens to structure S at a cold day? Structure S, we talk about vasoconstriction. We are saying S is the blood vessel, so it is called vasoconstriction. Vasoconstriction. That it actually shrinks in order for blood to flow on the deeper surface of the skin. So therefore, we can have less loss of heat from the what to the surrounding environment. That's what happens there. Give the name of layer Q and state two functions of this layer. It is actually the dermis. Then Q is actually the dermis. We're saying that the skin is dissected into three layers. So the uppermost layer, we say that is called the what? Is the skin, which we said it is the what? It is the dermis. It is the dermis, that one. So now, what is the function of the dermis? What is its function? It actually, we are saying, it actually, the functions one, it actually has blood vessels for temperature control. Has blood vessels for thermoregulation. It has got blood vessels for thermoregulation, and then it also has two function two. We are talking about the nerve endings for touch and pain. It also contains, it has got nerve endings for sensation. It has got nerve endings for sensation. We can talk about pain, we can talk about EG, we can talk about EG, we can talk about pain, we can talk about touch, or we can talk about pressure, such as those. We can give examples as many as we can. We can give so many examples that we can give there. Let's just answer the last question. The last question which we can answer here. Let's just go to the last question. The last question which we can answer here now is mammals are described as endotherms and reptiles are described as ectotherms. Distinguish between the two. I was saying the endotherms and exotherms. uh, what happens is that we need to understand that the endotherms and exotherms, in this case, we are talking about temperatures which can regulate their own temperature. I mean, animals, organisms that can regulate their own temperature. And then others can also not regulate, they can also control their environment from their temperature from the external environment. So what we need to understand is that these are just different differences that we can actually uh, draw. We can actually draw, and we actually have to be just easy. It is not, it's nothing much that is there that we can actually worry about. But in other words, we are just talking about the thermoregulation process. That is just that's what we just need to look at in terms of the temperature regulation. So when you look there, we are saying the endotherms, we are looking how they regulate their body temperatures in these two systems. So now, when we say the endotherms, the endotherms, the endotherms, they generate heat internally. Generate heat. They generate heat internally, and then they produce heat through metabolic processes, actually internally through their metabolic processes, through metabolic processes that are responsible for that, allowing them to maintain a relatively, the temperature is relatively stable. So we can talk about humans, and we can talk about examples of humans and birds. So what happens is that uh, it is actually controlled through shivering and sweating. E.g., we can talk about shivering, e.g., shivering or sweating. And then the ectotherms rely on external sources of energy, and and what we call ectotherms. Ectotherms, they actually rely on external sources of energy, rely on external sources of energy. They rely on external sources of energy. We can talk about e.g. The sun, e.g. sun. So in other words, they need a warm environment to regulate their temperatures, to regulate their body temperatures. And then it fluctuates. And what we mean is to say their temperature, their temperatures, it fluctuates according to the environment. Their temperature fluctuates according to the changes of the environment in terms of the ectotherms. Their temperatures fluctuate. They fluctuate according to what? According to the changes of the environment, according to the changes that are within the environment. They fluctuate with the environment. So that is the difference. So the endotherms, their temperature is more constant. They have got a more constant temperature. More constant what? They have got a more constant temperature. But now the ectotherms, their temperature is actually so dynamic. So in terms of the ectotherms, We have got examples there which we can say that reptiles and amphibians are examples in this case, e.g., e.g. reptiles. So now they fall into what? Into ectotherms. That's how we can classify, or that's how we can distinguish them. I'm gonna just make this one. An investigation was set up to observe plant growth patterns. We talk about tropisms, the tendency of plants to grow towards and away from a certain stimulus. We call that tropism. So two pods, P and Q, each containing three young plants of the same age and height, were placed inside a large cardboard box. A circular hole was cut in one side of the cardboard, and then this directional stimulus was placed at X. So this is what happened. And then the young plants had all been grown under the same conditions, but the young plants in port Q received additional treatment as the investigation was set up. So Q received what? Additional treatment. And then what happens is that the diagram shows the appearance of the seedlings after they have been in the box for 4 days. And then now identify the directional stimulus at X. You know, I'm saying when we look at this class, we are looking at the effect of what? The effect of various stimuluses that affects the plant growth. So in other words, what you need to understand is that when we are looking at this setup, it actually shows us that we are looking at tropism, which I'm saying we have got two types of them, geo and phototropism. Geotropism is on roots, that is the tendency of the roots to grow towards gravity. So in this case, this is light, which has got to do with the stem. The stem, it actually responds to what? To light. Light. We call that phototropism. Phototropism. So identify the stimulus. That one is what? Is it's actually phototropism. Phototropism. In other words, we call that the stem is positive. Phototropic. Positive. Phototropic. Is called positive phototropic. State the term used to give the growth response in this. State the term used to state the term used to give the growth response in this. So in other words, we are saying this: identify the directional stimulus placed at X. It is light. We are saying the first question. We are saying this is. We can say the first question is light. Is light, and then it is light, and then we are saying it is called positive phototropism. Light and the stem is growing towards light, and therefore it is called positive phototropism. And then name the plant hormone responsible for this response. It is auxins. Auxins are the ones that are responsible for this. Auxins, which are produced, we are saying auxins are produced at the tip, they are produced at the tip of the stem, at the tip of, the stem, or at the tip of the root, which we say they are produced at the fresh parts of a plant. That is one thing that we need to understand here. But the auxins are produced there. It is auxins that are produced there. So now what happens is that they actually accumulate, they are produced at the what? At the tip of the stem, and then that's what you need to to take note of. Explain how this hormone named causes this plant growth. Auxins are actually, what happens is that the stem bends towards light, and how this hormone named causes this growth response. So what happens is that in this case, there is a lengthy explanation anyway, but what you need to know is that auxins are produced. I mean, what happens is that light comes from the one, when a light is produced from one direction, auxins shun away from light. Uh, when auxins are produced, when auxins are produced, when they are produced at the tip of the stem, uh, they move away from light. They move away from light and then uh, hide at the dark side, dark opposite side of the stem. And then the opposite side, the dark side, it grows faster than brighter side. So uneven growth results in the stem bending towards light. So there is an even growth that is experienced by the stem. One side that is receiving light grows slower than the opposite side. So the dark side grows faster. As it grows faster, it therefore causes the stem to bend. Uh, it causes the stem to bend towards light because it is growing faster. Therefore, the stem will bend towards the source of light, which we talk about unilateral light subjection of a plant to what? To light. That's what we need to understand. From your observation of the diagram, suggest and explain why the plant in port Q do not show the same response as the plant in port Q. If we can look at this, in port Q, the plant does not change, does not bend. There is one thing that we can see. We are saying, as you can see here, Q, the apical buds are cut, apical buds are removed, and this one does P does have P is and P produces auxins, and then in this case there is absence. There is absence of auxins in what in Q. That is what is happening in this case. The reason is that uh, plants in Q do not produce auxins. The apical buds The apical buds are removed while the buds are present and auxins are produced. Auxins are being produced, causing the elongation of the stems. That is what is happening there. And then in modern agriculture, farmers tend to use synthetic plant hormones for herbicides. What are herbicides and what are they used for? Herbicides, these are chemical substances. These are chemical substances that are used to control weeds, that are used to control weeds. It's a chemical control mechanism. They are a chemical way in terms of weeds control. Chemical method of weeds control. So herbicides, they kill you, they kill weeds. So class, thank you very much for your time, and all the best in every other upcoming exams and so forth. I hope that you are going to have a nice revision. Until we meet again in the next videos. We are going to do paper 3 and paper 5. They are loading and coming. Thank you very much for your time. Let's just keep on working and doing this together. Don't forget to subscribe. You are at Metric Hub and it is there for you. Just know that quite a lot is loading. Don't repeat, don't fail. Let's continue. Let's do the best. All the best in every other exam. Let's interact here. Share the channel to others. Like and also turn on your notification button. Share and send. Friends and families, let them be connected. We're gonna do this together. Until we meet again in the next video. Keep on connected and following. Thank you very much.